Hello friends, welcome to Biology Extrema, where we teach biology from the basics to the extreme. Myself Keshav Bansal, and today we are going to study phospholipids. Okay, so what are these phospholipids? Okay, as we know, these are a type of lipids. Okay, because there is a lipid and there is something added in as a prefix to it. Okay, so it is a type of lipid. Okay, and we have already discussed the lipids, that lipids are amphipathic molecule. Okay, lipids are amphipathic molecule, or we can also say amphiphilic molecule. What do we mean by that? That it contains a polar group, okay, which is a hydrophilic group, water loving group, okay, hydrophilic, and it also contains a non-polar group, which is a hydrophobic group. Okay, usually we think that lipid is all about non-polarity, but no, it contains a polar group and it also contains a non-polar group. Okay, and that's why these kinds of molecules are called amphipathic molecules. Okay, so any type of lipid is gonna be an amphipathic molecule. Okay, and we have already discussed in the lipid video that lipids can be a simple lipid. There are actually various ways to define lipids based on the classification we are doing. Okay, but if we are talking about the simplicity of this structure, okay, if you are talking about the components, then lipids can be classified into simple lipids and complex lipids. Okay, and in simple lipids we have already discussed triacyl glycerols and waxes. Okay, now we have started this complex lipid part. Okay, and in complex lipid we have first this phospholipid. Okay, so as I told you that this simple and complex is based on the components. Okay, it is based on the components. So we will start with the component. Clear? So look at this diagram, that the phospholipid, okay, here it is classified into two types. We will look at it, okay, later on, but now for the moment focus focus on this just, okay, on these diagrams. Okay, so in phospholipid what we will found an main alcohol. Okay, we will found an main alcohol. Okay, let me just rub all this part, and we need to focus. Yeah, we will have this main alcohol. Okay, these are two examples we have here. We have alcohol. Okay, and to these alcohol what we have fatty acids attached to them. Okay, we have fatty acids attached to them. Okay, we don't need to focus on what kind of linkages and what kind of how many fatty acids are attached. Okay, we will discuss it later. Main thing you need to focus that the first component is this main alcohol, and then second component is fatty acid. Okay, so this is the alcohol and this is fatty acid. Now the third component is a phosphate group. Okay, a third component is a phosphate group. This phosphate group, let me take another color. This is a phosphate group. Okay, so and this phosphate group is directly attached to this main alcohol. Okay, fatty acids are directly attached to the main alcohol. Clear? Now this fourth component, which is also an alcohol, the fourth component is also an alcohol. It is usually an alcohol. We will see it later. Okay, in sometimes it can also be a water molecule. Okay, but in most of the cases it's an alcohol. Okay, so this alcohol is attached to this phosphate group. It is not attached to this main alcohol directly. It is first attached to this phosphate group. Okay, it is attached to this phosphate group. So as we can see, an alcohol which is attached to this phosphate group. Okay, and together these two forms what we call polar head group. Okay, polar head group. Okay, in this whole part only fatty acids are non-polar. If you want to classify which part is polar, because we told that it's an amphipathic molecule, so which part is polar and which part is non-polar. So this is the only part which is non-polar, which is hydrophobic. Rest of the part are polar. Alcohol will always be in polar molecule and phosphate is also in polar molecule. Okay, okay, so now we know the components of phospholipids. Now we'll start to classify it. Okay, so we are going to look at this main alcohol. We are going to look at the main alcohol to which fatty acids and phosphate group is attached. Okay, if the main alcohol contains glycerol, okay, we can get only two choices. Either it can be in glycerol or either it can be an sphingosine alcohol. Okay, glycerol is also an alcohol, which you already know. Okay, and sphingosine is another alcohol which you may not know up till now. Okay, but it is a type of alcohol. Okay, so but we have only two options in the case of phospholipids. Either it can be glycerol or it can be sphingosine. Okay, so remember that. If it is glycerol, then we call it glycerophospholipids. If it is sphingosine, we call it sphingophospholipid. Okay, and what make them phospholipids? Okay, they can, because due to the presence of this phosphate group, it will always be the common thing. Okay, because due to these are categorized into phospholipids. Okay, now we're going to talk about the bonds. Okay, let's say start with the glycerophospholipid. Okay, so in glycerophospholipid, these fatty acids, okay, these fatty acids, let's say we have 3 carbon, and we have actually 3 carbon, the glycerol, carbon 1, carbon 2 and carbon 3. Okay, to the carbon 1, the fatty acids, and carbon 2, the fatty acids are attached with the glycerol. This linkage, this bond is called ester linkage. Okay, this is called beta ester linkage. Clear? While this polar head group, as I told you that we can use the term polar head group. Okay, if you are talking about both of them in a combination, then this polar head group is attached to the glycerol molecule through phosphodiester linkage. Okay, phosphodiester linkage. Clear? So let's look at it. So as I told you that based on the alcohol we used, based on the main alcohol, glycerol is sphingosine, it can be phosphoglyceride, or see, these are the two names of the same thing, phosphoglycerides or glycerophospholipids. Okay, while the other one where sphingosine alcohol is present, then it is called sphingophospholipid. Now we're talking about phosphoglycerides or glycerophospholipids, in which main alcohol is glycerol. So it has these components. Okay, nothing of any surprise. The main thing is that now we'll talk about this another alcohol. Okay, we'll talk about it in a moment. Let's look at what we have studied, that the two fatty acids are attached. These two fatty acids are attached with the glycerol molecule with the help of ester linkage, while this polar head group, okay, which consists of a phosphate and an alcohol group, is attached to the third carbon of the glycerol with the help of phosphodiester linkage. Okay, you don't need to remember first and second and third carbon, you need to remember the linkage. Clear? And also here it is mentioned that that phosphoglycerides are the most numerous, most abundant cell membrane lipid molecule. Okay, this is part from cell biology. Okay, let's have a look at it. If actually what happens, if we are talking about cell membrane, or we can say plasma membrane, cell membrane, plasma membrane, so what happens is that if I talk about how many lipid molecules are present in the cell membrane. Okay, so we can categorize into major 3 types. The first can be a phospholipid. Which we are discussing. Okay, but there are 2 other categories also. It can be a glycolipid. Okay, and it also can be an sterols. Okay, sterols like cholesterol, ergosterol, cetosterol, sigmasterol. Okay, depending on the type of organism we are talking about. So these 3 membrane lipids are present in the cell membrane. They can be present in the cell membrane. Okay, they actually are going to present the cell membrane in every case. So what happens, that if I ask you among these three, which membrane membrane lipid is the most abundant one, so the answer is going to be phospholipid, and this is asked in the MCQs. Okay, the phospholipid is the most abundant lipid in the cell membrane. Okay, now we'll talk about it in a moment, that there can be different type of phospholipids also. Okay, based on the other alcohol which is attached to the phosphate group. Okay, and then we can ask that among these phospholipids, okay, or we can add some options other like cholesterol also, or other lipids molecule. Then we are going to ask that which kind of lipid membrane lipid molecules are present in the cell membrane. Okay, so we know that phospholipids are the most abundant, but in phospholipids we can also ask which type of phospholipid is more abundant particularly. Okay, so let's talk about this. As I told you that the main alcohol can only be glycerol, or it can only be an alcohol sphingosine, but this another alcohol of polar head group, it can be of different types. Okay, it can be of different types. So let's look at it. In the most simplest case, it can be water too. Okay, it can be a water too. So if it is used as water, in most of the cases it will be a alcohol. Okay, but it can be a water too. Okay, and if water is used, it will be the simplest glycerophospholipid, and we call it phosphatidic acid. Okay, we call it phosphatidic acid. Okay, and if we have used choline here, okay, let's look at this diagram. Okay, if we use the water molecule, what we will get from this? We get an hydrogen and OH group. So hydrogen, okay, so hydrogen gets bind to this. So here X part is hydrogen, which comes from water. Then in this case, the phospholipid is the glycerophospholipid we are talking about is a phosphatidic acid. Okay, this is the component we have already talked. This is the glycerol. Okay, these are the two fatty acids. Okay, and the fatty acids can be both saturated or unsaturated. Okay, and we have already made a separate video on fatty acids. You can watch in the playlist. Then there is, we told you that there is polar head group. Okay, this is the polar head group, and in this we have a phosphate group and a head group substituent, which is usually comes from an alcohol like choline, ethanol, amine, serine, but it can come from water too. Okay, it can come from water too. So if it is if this head group substituent come from the water, then it is called phosphatidic acid, which is the simplest type of glycerophospholipid. And if there is this alcohol like choline, then we will call this head group phosphocholine, because it is the choline part. Okay, this is the choline part, and with the phosphate group, it forms phosphocholine. And if I name this glycerophospholipid with a phosphocholine in it, then I will say phosphatidylcholine. Okay, you are getting me? Okay, let's take if I use serine. Then this X part comes from the serine alcohol. Okay, and if I name this head group polar head group, then I will say phosphoserine. Okay, if I use ethanolamine, I will say phosphethanolamine. But if I give the name of this complete glycerophospholipid, okay, then I say phosphatidylserine or phosphatidylethanolamine. Okay, and also there are other common names which are asked in the exams. Okay, because these are common name. Okay, you can recognize by these, but these you need to remember: these lecithin, cephalin, cardiolipin. And there's an interesting note to take here, that in the case of cardiolipin, the cardiolipin are the phospholipids which are present abundantly in the inner mitochondrial membrane, which makes it more impermeable in comparison to the outer membrane of the mitochondria. Okay, you will study in the cell biology part in the mitochondria. There it will be mentioned that inner membrane is more impermeable in comparison to the outer membrane. Outer membrane is more permeable due to presence of transport proteins called porins. Okay, mitochondrial porins. But inner mitochondrial membrane is more it is actually impermeable. Okay, due to the presence of cardiolipin. And now we know this cardiolipin is nothing but diphosphatidylglycerol, okay, a kind of phospholipid, in which polar head group we use phosphophosphatidylglycerol, okay, you getting me? And these are the net charge which can be present. This is also important, okay. So we have discussed glycerophospholipid, and now we need to come on sphingophospholipid, okay. Then we will come to this third type. So in sphingophospholipid, we know that the main alcohol we use is sphingosine, and to this a single fatty acid is attached. In the case of glycerophospholipids, as we can see in the figure, we have two fatty acids attached here, okay. We have two fatty acids attached in the case of glycerophospholipid, okay, and these are attached with the glycerol with the help of ester linkage, okay. But in case of sphingophospholipid, a single fatty acid is attached with the sphingosine alcohol with the help of, now this is important, amide linkage, not ester linkage, okay, it is amide linkage, okay. And together, when a fatty acid is combined with the sphingosine alcohol, we can use a term called ceramide. Okay. So look how examiner take advantage of this. That if I ask that what if I ask you about the components of a sphingophospholipid, okay, and there is a common example of sphingophospholipid which are going to discuss, which is called sphingomyelin. Sphingomyelin is an example of sphingophospholipid, in which we use an alcohol called choline, or we can use ethanolamine, okay. Based on that, it is specified into sphingomyelin. There can be other alcohols which will form other types of sphingophospholipid, okay. So in case of, let's say I am asking about the components of sphingophospholipid or sphingomyelin, okay. So I can, one way to ask is that I can say that is plus sphingosine alcohol plus fatty acids plus phosphate group, or I can combine called phosphocholine or phosphoethanolamine, okay. One way to give an option, correct option is this, okay. But if I am trying to trick the student, I can ask, because I know that sphingosine alcohol, when attached to a fatty acid, it forms ceramide, okay. So now I can ask in components that ceramide plus, let's say, phosphocholine or phosphoethanolamine, okay. Now student will think, let's say if I provide only this option, the student will think that all the options are incorrect, okay. If I provide this ceramide plus phosphocholine and rest, this is actually correct, and if I provide other options, they will be incorrect, okay. So student will get confused that which option is correct, because there is no mentioning of sphingosine, there is no mentioning of fatty acid. But so you need to remember that ceramide is sphingosine plus fatty acid attached by an amide linkage. This is very important, okay. And the polar head group is attached with the sphingosine alcohol with the usual phosphodiester linkage, okay. This is the same. Clear? So the fatty acid, it can be saturated or it can be monounsaturated, means only a single double bond is allowed if it is to be unsaturated, okay. It cannot be a polyunsaturated. This sphingomyelin, it can be present in plasma membranes of animal cells and in the myelin sheath of nerve axons, okay. This is important, this can be asked. And the alcohol in the polar head group is or we can say the polar head group can be phosphocholine or phosphoethanolamine, okay. There can be other types of alcohols in case of other sphingophospholipid. Here we talked about specifically sphingomyelin, and sphingomyelin is all you need to focus on in the case of sphingophospholipid, okay. Now we have third type of phospholipid, which is called ether glycerophospholipids, and this is very important because it is asked in the case of RKE, okay. In RKE we have two fatty acids like in the case of glycerophospholipids, okay. But the first fatty acid is attached with the glycerol molecule with the help of ether linkage, okay. Ether linkage. not ester, and only the first fatty acid. Okay, and the second fatty acid will the second component will be attached with an ester linkage. Okay, then there also will be phosphate group and alcohol, okay, which is attached to with the help of phosphodiester bond. The main defining character of ether glycerophospholipids is that there is ether linkage between fatty acid and carbon 1 of glycerol. Okay, so let's look at it. Ether linkage is there. Okay, the main two common examples are platelet activating factor, okay, and plasmalogen. Clear? Here is the diagram. This is the diagram. Okay, so this is this first is the ether. This is the fatty acid which is forming ether linkage. Okay, it is plasmalogen. It is platelet activating factor. Clear? So this is all about phospholipids. I hope you understand the video. If you understand the video, like it, share it, subscribe our channel, and if you can, please donate a small amount. If it will help us in providing this noble service of free lectures and quality notes. So small help from your side creates a huge difference. Okay, so thank you from Biology Extrema. See you next time. Take care.